Welcome once again to Professor Kareem Ainsley Encounter. Today we'll be examining the fundamentals of immunology as it surrounds the concepts of blood banking. In this session, we'll be diving deep into the immune system, innate immunity, acquired immunity, cellular and humoral immunity, cell organs such as B cell, T cell, antigen-presenting cell, immune system organs, immune maturation, cell lineage and markers, cytokines, and immune system genetics, characteristics of the immunoglobulin, which will be a fun area to examine. We will be examining the structure, the immunoglobulin significance for blood banking, the variants, the FC receptors, and we'll also be discussing complement system, the classical pathway, the alternative pathway, the lectin complement pathway. Then we'll be looking at the membrane attack complex, binding of complements to the RBCs and antibodies. We'll examine in part the characteristics of the antigen, characteristics of blood group antibodies, and then we'll be doing the antigen-antibody reaction, detection of these red blood cell antigens and antibody. We'll most certainly be looking at the factors that influence agglutination reactions such as centrifugation, antibody ratio, effect of pH, temperature, immunoglobulin type, the enhancement media, the protein media, the low ion strength solution media, polyethylene glycol and polybrine, proteolytic enzyme, anti-human globulin reagent, chemical reduction of IgG and IgM molecule. So we have quite a bit to cover. And not to mention, we will also be looking at blood products, transfusion, and the immune system. If this is something that you're interested in learning about, please join us as we get on the inside. We'll have time signatures and a detailed description of different areas, and our resources can be found in the description below. So please navigate to the description so you can find the resources that we have used and we would recommend it. Information presented here is absolutely free for your listening and visual pleasure. Let us get in. As it relates to the immune system, it is a complicated, tightly controlled, and includes tissues, organs, cells, and biological mediators that coordinates to defend a host organisms against intrusion by the foreign substances or abnormal cells of self-origin. So in this case, we're speaking about it protects the body, the human body, or whatever host body from invading pathogens and also from self-intruders such as autoimmune cells which fail to respond to apoptosis. Immunity refers to the process by which a host organism protects itself from attacks by the external and internal agents. So you can have attacks coming from outside like virus, fungi, bacteria, etc. And the internal agents such as inflammatory cells that develop based on carcinogenic exposure and autoimmune conditions that may be activated, among other variants. Immunity also confers the protection for non-self and abnormal self-element, which are controlled at different levels. So we have different levels to this whole system of immunity and the immune system. So you have the first door then you have the second door, and you even have a third door. And all these doors work together as protectors. It's like having a army, and you have the front line, those in the middle, and those at the back of the team. The number of different types of non-cell organisms include unicellular and multicellular organisms such as viruses, bacteria, mycoplasma, fungi, and parasites. And additionally, sometimes we have tumor cells and cells destined for termination within the host. They need also to be recognized so that they can be eliminated. And this is why the immune system is so important, because if it had not been fully functional, then the human body would be compromised. This response and elimination of organisms and unwanted cell is accomplished through cellular and or humoral mechanism. When we speak of the cellular defense mechanism, this is mediated by various cells of the immune system, such as the macrophages, the T cells, and the dendritic cells, which function to eliminate virus, bacteria, cancer cells, and other cellular pathogen. Then we have the humoral mechanism, which comprises of the B cells and the specific antibodies they produce, along with complement complexes. When we speak of the humoral mechanism, this comprises of the B cell and specific antibodies they produce, along with complement components that are produced in the plasma, saliva, and other body secretions. So these antibodies or immunoglobulins then bind to specific receptor sites on the cell. The extent of the damage that is done to the target cell is dependent upon the complement pathway that is activated and several other host factors. Antibodies or immunoglobulins have a special significance for transfusion medicine because antigens present on the transfused cell may cause reaction in the recipient and complicate the therapy because antigen tend to find antibodies, and if a complex is formed, then that there is considered an agglutination, and when agglutinations occur, then it puts the body in a state of panic, which can lead to transfusion reaction and graft versus host disease state and rejection. We may even have graft rejection and all manner of complication that may become evident. The majority of the blood bank testing is focused on the prevention, the detection and identification of the blood group antibodies and on the typing of the red blood cell antigen so that they can have an idea as to what antibodies are present and what antibodies should this individual be staying away from. Understanding the immune system and its response is essential in understanding the factors that can affect agglutination reaction between the red blood cell and the antibodies and various testing modification. So we can detect alloantibodies or autoantibodies in routine blood bank testing procedure. The detection is dependent on several factors, including the binding force between the antigen and antibody, the property of the antibody itself, and the individual characteristic of the host. Antigen-antibody reactions are influenced by a number of factors, including the distance, the antigen-antibody ratio, whether it's pro-zone, post-zone, the pH, whether it's acidic, alkaline, temperature, whether it is 37 degrees, which is the body temperature, or whether it reacts at a lower temperature, and the immunoglobulin type, whether it's IgG, IgA, IgM, IgE, IgD. A host of factors can affect the whole immune system and its response. But we're going to simplify all of these chasms that surround the immune system so that we can all have a better appreciation as to the functionality of the immune system as it relates to blood banking. All organisms are from time to time challenged by the potential of harmful external and internal organisms or molecules from their environment that they must be protected against. One of the most fundamental concepts in immunology is the idea of self versus non-self and how the immune system distinguishes between the two. When we consider self, we refer to anything that is derived from the host genome and the rearrangement of the host genes. It includes cells, fluids, molecules, and more complex structures of the host organisms. Anything put into the host body, even a close genetic match, can be recognized as non-self and therefore can be rejected by the response of the immune system. It includes cells, fluids, molecules, and more complex structures of our host organisms. So anything put into the host body, even a close genetic match, can be regarded as non-self and therefore can be rejected by a response of the immune system. And in this case, we can have an idea as to how organ rejection takes place. So though it may be a close genetic match, it's not necessarily a perfect genetic match. And that is why there is a likelihood of the immune system recognizing the replaced organ as non-self. And for this reason, suppressants are normally given in order to maintain the organ's viability. Now, let us move into non-self. Non-self refers to anything physically outside of the host, whether a living organism such as the parasites, the fungi, the bacteria, or non-living toxin such as a poison ivy fluid, insect venom, carcinogens, and a host of other agents that can have adverse effects on the organism. Now, when foreign objects or damaged host cells are detected by the immune system, an immune response occurs because there is a detection of something being wrong, and then these soldiers decide that, hey, it's time for us to go and defend. So that is the mounting of the immune response. The immune response occurs at different levels and are either the primary, the natural immunity, innate immunity, or the secondary, adaptive or acquired immunity. The innate immunity consists of physical barriers, biochemical effectors, and immune cells. The first step of innate defense is external, which includes our skin and enzyme present on the surface. The second line of the innate defense is internal and can recognize common invaders with a non-specific receptor, such as phagocytosis that does not have to be primed. The last line of defense is acquired immunity, which needs time, days to weeks, and reorganization to mount an effective and a specific reaction. Importantly, it is acquired immune response that respond more rapidly and efficiently against a repeat attack from a pathogen by establishing the memory response. So yes, the immune system has memory and it has memory cells. Immune system specificity also prevents the host from becoming attacked and damaged during an immune response, because sometimes we have invaders which come into the host looking like the host cells, and they create or allow the body to mount a response. So for the safety of the host for self-preservation, the immune system has to be so specific that it does not destroy the host instead of the lookalike. Another mechanism is the localization nature of an immune reaction, which also prevents systemic damage throughout the host organism. So the host tries to send all the fighters to the area which is injured and try to curtail everything within that area, so that the blood system does not become contaminated with the invader. So they try to restrict the movement, just like when we have zones of special operation, or when we have curfews, or when we have barriers set up. The immune system operates on similar principles. So both the innate and acquired immunity are two arms of the immune system, and they work to prevent infections and damaged cells from destroying the host. The innate part of the system is more primitive and does not function in a specific way. Rather, it recognizes certain complexes, repeating patterns presented on common invading organisms. The innate arm can function immediately to stop host organism from being infected. Therefore, the innate immune system is immediate. It is the first line of defense from invading pathogens. While on the other hand, the acquired immune system is said to be a little bit more advanced. It relies on the formation of specific antigen antibody complexes and specific cellular responses. Acquired immunity allows for a specific response, and immune system memory allows resistance to pathogen that was previously encountered. So the immune system is working from the innate and the acquired state, and both are working together to protect the host organism. Now, let us look more in-depthly at the innate immunity. There are two important features of the innate immunity. First, the innate immunity system is non-specific, so we have to get that out of the way. It is not specific to any particular invader. It just identifies something that is not self, something that is an invader, and then it just attacks. The same response is used against invading organism, no matter what the source is, as long as the immune system can recognize it as non-self. The innate immunity is present at birth and does not have to be learned or acquired. So now the second thing that we need to note is that it does not need modification to function, and it is not altered with repeated exposure to the same antigen. Because the innate immunity functions so well as a first line of defense, the innate immune system is comprised of physical and biochemical barriers as well as numerous cells. Let us look at some of the physical barriers, which includes the intact skin, not the broken skin, because if the skin is broken, then it means that invaders can enter. An unbroken skin is a first line. Then we have the mucous membrane. The mucous membrane is there to trap anything that is coming in. We have the ciliary lining. The ciliary lining is supposed to push out whatever foreign bodies or debris are trying to make entry in. And then we have the cough reflex. The cough reflex is that if something funny goes down or something that tickles and it needs to be removed, we can just cough it up and carry it right back up. Then we have the biochemical barrier of the immune system, which includes the bacterial enzymes such as the lysosomes and the RNase, the fatty acids, the sweat, the digestive enzymes in saliva, the stomach acid, and the low pH. So we can understand the lysosome enzyme and the RNase, the fatty acid. We understand the sweat that push out the excess from the pores, the digestive enzymes which breaks down the food and destroys some of the pathogens. Then we have the stomach acid, which is so intense that most organisms cannot survive in it. And we have the low pH. The innate immune cells include the phagocytic leukocytes and the natural killer cells. Yes, we have the natural killer cells in the innate immune system. The phagocytic cells of different types are found in most tissues and organs of individuals, including the brain, because the brain is so important. It has to be protected. A brain damage can destabilize a human and can cripple all other bodily function. The liver, the intestine, the lung, the kidney, all of these are major organs which are needed to work at full capacity in order to have a healthy individual. Phagocytes such as circulating monocytes in the blood and the peripheral macrophages can move between vessel wall, which is a plus. So instead of going around a long way, they can just go through the vessel walls and take the shortcut. That's how I want you to look at it. And these are the monocytes that we're speaking about in the blood and the peripheral macrophages, which can move between the vessel wall. Phagocytes can recognize complex molecular structures on the surface of invading cells or in the secretion and the fluid of the host body. They remove the invading organism by engulfing and digesting them with visceral enzyme. Two major cells that can use phagocytosis to remove pathogens are the polymorphonuclear cells, which include the neutrophil, the basophil, and the eosinophil, and the mononuclear cells, which include the monocytes in the plasma and also the macrophage in the tissue. Opsonins are the factors that include the antibodies and the complement components in the plasma that coat the pathogen and facilitate phagocytosis. In order for the cells to be marked for phagocytosis, they have to be coated so that the body can recognize. When phagocytes ingest the foreign cells and destroy them, they can be activated to release the soluble polypeptide substance called cytokines that have numerous effects on other cells of the immune system and the vascular system. So these two systems have to work in tandem with each other. The vascular system to increase blood flow or to decrease blood flow from a particular area, the immune system to send the prerequisite cells to the various area, and also the cytokines help to regulate the immune response in terms of specificity, intensity, and the duration. Another important component of the innate immune system is the complement system. The complement system has three major roles in immunity. The first, the final lysis of abnormal and pathogenic cells by binding of antibody. The second, opsonization and phagocytosis. And the three, to mediate inflammation. So these are three critical areas that are necessary for the immune system to work. These are important components of the immune system. So now that we are talking about complements, the complement proteins need to be activated. And there are three pathways that are available for the activation process. We have the classical pathway, and then we have the alternative. We have the lectin pathway as well. So those are the three major pathways. The classical, which is standard. Then we have the alternative. If one doesn't work, then we can move to another. And then we have the lectin based on the different pathogen that are being safeguarded against. The classical pathway uses antigen antibody binding and therefore is a specific activator of complements. The alternative pathway activates complement by recognizing polysaccharides and liposaccharides found on the surface of bacteria and the tumor cell. Therefore, it uses non-specific methods of activation. The lectin pathway is activated though by the mannose binding protein bounded to the macrophage. Inflammation is initiated by any type of tissue damage. So burns, infection, fractures, necrosis, and superficial wounds all elicit an inflammatory response that is characterized by an increase in blood flow to the wound area, increasing blood vessel permeability at the site to allow a greater flow of cells and mobilization of phagocytic cells into the site, and a possible activation of acute phase and stress response protein at the site of the tissue damage. Eventually, the wound is repaired, new tissue grows in the place of the damaged tissue, and the inflammation stops. So the body is operating as a pre-programmed machine. You don't have to tell the body that, hey, you need to activate phagocytosis. The body already comes pre-prepared, knowing exactly what needs to be done. And that is the beauty of the immune system. Now that we have covered the innate immunity, I think it's time for us to move over into the acquired immunity as we continue our exploration. Acquired immunity is another major arm of the host immune system. It is most specific and allows the immune system to have memory of pathogens it has encountered previously. This acquired system is present in vertebrates. As we examine the term acquired basically refers to the immune system having garnered knowledge of or the fact of a previous encounter is acquired, developed by specific contact with pathogen or aberrant cell. The term adaptive refers to the ability to change, to modulate, and destroy new complexes of pathogen, although it must first react to them through the complex recognition process. Acquired immunity is specific in recognition of new pathogens and has specific responses depending on the type of pathogen that it encounters. So this type of immune system uses antibodies as a specific immune effectors. Antigen specificity and uniqueness determines the particular antibodies that will bind to it. The antigen-antibody complex is a three-dimensional interaction, which helps to prevent the binding and induction of an immune response by non-specific antigens. For example, antibodies against one blood group's antigen do not react against another blood group antigen. An antigen that an antibody is made against is sometimes referred to as its antithetical antigen. Now, let us look at cellular immunity. Cellular immunity is mediated by various immune system cells such as the macrophages, the T cell, and the dendritic cell. Lymphokines are powerful molecules that includes the cytokines and the chemokines. These effectors molecules play a critical role in the cellular immunity by activating and deactivating different cells and allowing cells to communicate through the host body. Humoral immunity, though, consists of a fluid part of the immune system, such as the antibody and the complement components found in the plasma, the saliva, and other secretions. One of the most important part of the humoral immunity is the antibody, which is produced by the B cell. Antibodies are called immunoglobulin, immune because of their function, and globulin because of the type of globular soluble protein that it comprises of. They are found in gamma globulin proteins of plasma or serum. When it is separated by fractionation or electrophoresis, the function of the antibody is to bind to foreign molecules called antigen. Most antigens are found on the surface of foreign cells or on damaged self cells. A key feature of antigen-antibody reaction is the specificity. Only one antibody reacts with one antigen or one part, an epitope or an antigenic determinant of a complex antigen. An immune reaction against an antigen stimulates the production of antibodies that will match the epitope of the antigen. Then the binding reaction of antigen and antibody has often been looked at as a lock and key mechanism, referring to its specificity in conformation. Antigen-antibody complex formation inactivates the antigen and elicits a number of complicated effector mechanisms that will ultimately result in the destruction of the antigen and the cell to which the antigen is bound. And it brings us to the point that the study of antigen and antibody reaction is called serology. So in blood banking, serology is also being performed for most of the antibody screening methodologies, such as the indirect tests, the cross-matching techniques, and antigen-antibody complex screening in the plasma. The direct antiglobulin test relies on the detection of antibodies and complements bounded to the surface of the red blood cell. So yes, in immunohematology or blood banking, serology is also being performed. And that is why the profession of medical laboratory science, transfusion medicine, or even laboratory medicine is so intertwined with the whole functionality of the human body. Let us now look at cells and organs of the immune system. The different types of the cells within the immune system can be distinguished by the membrane markers they possess. These are referred to as a cluster of designation, the CDs. not to be mistaken for certificate of deposit, but they are the cluster of differentiation markers and are detected by the immunotyping method to identify cells of the immune system. Lymphocytes, which are critical for acquired immunity, are divided into two major types, the T lymphocytes and the B lymphocytes. The T lymphocytes mature in the thymus T lymphocytes, thymus T. The T lymphocytes mature in the thymus gland and is responsible for making cytokines and destroying virally infected host cell. The B lymphocytes mature in the bone marrow, B lymphocytes, bone marrow, and when stimulated by the antigen evolves into plasma cells that secretes antibodies. Natural killer cells are another type of lymphocytes that play a role in the immune protection against a virus. Dendritic cells are present throughout many systems of the body and are responsible for antigen processing and presenting. Dendritic presenting. Lymphocytes recognize only one specific antigen, which is determined by the genetic programming of the lymphocyte. Gene cell undergoes gene rearrangement in order to have the correct antibody made that can react with the correct antigen, because they are trying to elicit an antigen antibody complex so that phagocytosis can take place and the threat can be eliminated. So because we have so many antigens that are pathogenic, the immune system has been adapted to recognize millions of different antigens with specific response that will match only one particular antigen. Therefore, if a foreign antigen is present, only a small percentage of the host antibody and the T cell receptor will recognize it. In cases, though, where an antigen is recognized by more than one antibody, a process of clonal selections happen, in which the different cells that recognize a different epitope of the antigen are expanded. The mature B and T cells occur after an antigen is encountered. Both the B and the T cell mature into what we call the effector cell, which are the functional unit of the immune system. The final effect that the B and the T cell can induce is the elimination of a pathogen and foreign cell. Immune memory is also acquired when the lymphocytes mature into memory cells after antigenic stimulation, thus allowing the immune system to recognize antigens from previous encounters. Once the infection is cleared, some of the cells that can recognize the antigen remain in circulation. The lymphocytes are then permanently set to respond to the same antigen again by a process of memory. The second time this antigen is encountered, memory allows for a more efficient and rapid immune response because they already know it, the targets have been identified, and they already have a copy, so they just multiply the copies. They don't have to start again from scratch in creating the defense. Let us examine the B cells. Antibodies can be secreted or membrane bounded, but are produced exclusively by mature B cells called plasma cell. So mature B cells are called plasma cell and they produce the antibodies. The receptor of the B cell that recognizes the antigen is a membrane-bound antibody. When the receptor antibody on the B cell reacts with a specific antigen and recognizes it, the B cell is activated to divide. So it's a cascade of reactions that are taking place in order to activate the immune response. The cells produced from this rapid division matures into plasma cell and memory B cell. The memory B cells have antibodies on the surface that is of the same conformation from which they were divided. The plasma cells are antibody factories that make a large amount of a specific type of antibody in a soluble form that remains in circulation in the plasma and body secretion and also the lymphatic system. Antibodies can neutralize toxic substances and antigens that are encountered by binding to them and thus prevent them from interacting with the host. We don't want them to react with the host any at all because that could compromise the organism. When the antigenic sites is non-reactive because of antibody binding, it cannot interact with the host cell to infect them or damage them. So they have been debilitated. Also, binding of antigen by antibody brings about opsonization, which aids in the direct killing of the pathogen by cell lysis. Now, let us look at the T cell. The T cells are another important component of the cellular part of the acquired immunity. One of the major differences of the cellular immunity compared to the humoral immunity is that the T cell recognize antigens that are internalized within a host. The antigens are then processed and presented on the host cell's surface in small peptide fragments. The T cell mediated immunity is involved in the response against fungal, viral, and viral intracellular parasites. T cell mediated immunity is involved in the response against fungal and viral infections, intracellular parasites, tissue graft, and tumors. So the T cell receptors do not recognize foreign antigen on their own, as the B cells do. They require help in the form of cell membrane protein known as the major histocompatibility complex, the MHC molecules. The MHC genes determine the human leukocyte antigen HLA present on the leukocytes and other cells. The HLA molecules have been known for many years to be a major determinant of tissue graft rejection. And this is one of the things that we have to be very mindful about, especially when there is a transplantation and grafting taking place. There are two major classes of the MHC genes and antigens: MHC class 1 and MHC class 2. MHC class 1 antigens are found on most nucleated cells in the body, and MHC class 2 antigens are found on most antigen presenting cells. In humans, MHC class 1 codes for HLA-A, HLA-B, and HLA-C. So that is HLA-A, HLA-B, and HLA-C antigen, whereas MHC class 2 genes code for HLA-DR, HLA-DQ, and HLA-DC. The two major functions of a T cell are: one is to produce immune-mediated substances such as cytokines, which influences many immune functions throughout the body. The second function is to kill cells that contain foreign antigen. When T cells are activated by antigen, they start to secrete cytokines and change their cellular interaction. The T cells are also grouped into two major categories with two major functions. So we have the T helper cells, which is denoted by TH. Then we have the T cytotoxic cells, which is denoted by TC. So the T helper cells are also distinguished by their cell receptor, the CD4, and T cytotoxic cells are distinguished by the CD8 receptor. The T helper cells are further grouped into TH1 and TH2 and respond to different cytokines. The T helper cells have the ability to recognize antigen along the MHC class 2 molecules and to provide help to the B cell to evolve into a plasma cell and make antibodies. So it is the T helper cell which determines which antigen becomes immune system targets, as well as which immune mechanism will be used against them. These T helper cell aids in the proliferation of an immune cell after they encounter an antigen. We also have some cells called the APCs, which are the antigen presenting cells, and they are leukocytes, which include the macrophages, the neutrophil, and some B cell. In addition to the leukocytes, they are specialized immune cells capable of antigen presentation. These include a different type of dendritic cells presented in the skin, Langerhans cell, the nervous tissue, glial cell, lymphatic nodes, spleen, intestine, liver, the Kupffer cells, the bone, the osteoclast, and the thymus. These antigen presenting cells first phagocytize the foreign antigen, processing it internally, and with the help of MHC molecules, present short peptide sequences of the antigen on their cell membranes. T helper cells can then recognize the antigen in context of MHC presentation and respond to it by the appropriate immune reaction. The organs of the immune system are divided into the primary and secondary system. The primary lymphoid organs are the thymus and the bone marrow, where immune cell differentiates and mature. The secondary lymphoid organs include the lymph nodes and the spleen, in which the immune cell interacts with each other and process antigen. It usually takes days to months for all aspects of an efficient immune response to happen. From the moment of an intruder alert, an antigen is first encountered. The lag phase is until an appropriate immune response occurs is called the latency, or the pre-seroconversion, or the window period. It is during this time that the antibody cannot be detected with serological testing. During the latency period, however, the T cells and the B cells are very active in the process of processing antigen and initiating the primary response to the antigen. The first antibodies made against the new antigen are different from the antibodies of the secondary response. The primary antibodies are the immunoglobulin M subclasses, where the antibodies of the secondary response are the immunoglobulin G. So the primary is M and the secondary is G. After the antigen is cleared, memory cells are stored in the immune organs of the host. When the same antigen is encountered again, the memory cells are activated and produce a stronger and more rapid response. IgG antibodies are formed during the secondary response and are made in a greater quantity. And although IgM is made during the primary response, there is a period in which it overlaps with the production of IgG antibodies at the beginning of the second response. IgG secondary antibodies have a higher avidity for antigens and can be produced by much lower concentration of antigen. Secondary antibodies can usually be measured within 1 to 2 days. Let us now look at cellular lineage and markers. Nearly all cells of the host body, especially the various leukocytes, have specific receptors on the cells, CD markers that interact with other cells and receive signals from cellular messenger system. Macrophages, natural killer cells, the T and the B cells, and not to mention APC, antigen presenting cells. They can interact directly with the cell-to-cell communication or indirectly with soluble mediators through a complex system. These markers on cells can change during the lifetime of the cell or in response to an infection. Yes, that is the adaptive nature of these cells, or when they are activated. So currently, there are more than 200 different CD markers known. CD markers are critical to identifying hematopoietic cell maturation stages and lineages, especially when cell identification processes are being engaged. So what we need to remember and realize is that all immune cells originate from the pluripotent hematopoietic progenitor, or the CD34 positive cell, through one of two pathways of lineage. We have the myeloid or the lymphoid. And these two words will be important when we try to differentiate T cell, B cell mediated cells, and also when we try to identify different types of leukemia when we move into the hematological section of our discourse. But what we need to realize and recognize in this stage of our development is that growth factors are responsible for the differentiating and maturing of the stem cells into the many different cells of the immune system. So growth factor, very important, not only to just grow the human being, but also for cell proliferation. It is also necessary, and for the reproductive cycle of cells to occur naturally. Growth factors are also known as progenitor stem cells to reproduce and differentiate. Growth factors are also known as progenitor stem cells, and these should be associated with reproduction, reproducing, and differentiation, creating a distinction. Now, let us look at the myeloid lineage of cells. These cells consist of the phagocytic cells, such as the monocytes and the macrophages, also referred to as the mononuclear phagocytic cells, or the MPS for short. The granulocytes of the polymorphonuclear cells, PMNs, the neutrophils, eosinophils, and basophils, and also the antigen-presenting cells, the APC, such as the dendritic cells in the skin and the liver. Erythrocytes and platelets originate from this system. It is the lymphoid lineage, which consists of various subpopulations of the lymphoid cells, including the T cell, the B cell, and the natural killer cells. The myeloid monocyte precursors originate in the bone marrow and then differentiates into circulating blood monocytes. When the monocytes encounter antigen, they are differentiated in the tissue macrophages. Phagocytic cells, they process antigens from the acquired immunity, and they can directly kill many pathogens such as bacteria and fungi as part of the innate immunity or the innate immune system. The MPS cells present antigen to the lymphocytes and interacts with other immune cells via cell membrane receptors. The FC part of the antibody receptors and the complement receptor, the CR1, are used by the phagocytes during opsonization. When these cells are functioning as antigen-presenting cell, they express the MHC class II molecules on their membrane and lack the FC receptor. Now, let us look at granulocytes. Granulocytes originate in the bone marrow. They are the predominant leukocytes in the circulation of the mature adult. That accounts for about 60 to 70%. Granulocytes all have granules. That's why they are called granulocytes in their cytoplasm, and are of three main types, distinguished by the hematological staining of their granule. So the neutrophils stain a faint purple or neutral color, neutral granules. So neutrophil, neutral granules, gives us an idea as to how we can remember. The eosinophils are red or reddish orange. Eosin. Think about the eosin stain. What is coming to mind? Eosin. So it is reddish, and we can associate this with acidic granules. And the basophil, basophil, basic. Okay, so now the basophil are a bluish black color. So it gives us the idea: neutrophil, neutral. Eosinophil, we are thinking about acidic granules. Then when we go into the basophil, we are thinking about basic granules. So these are some ideas of how we can remember and recall as needed. The main roles of these cells are in the process of phagocytosis. They function primarily in the acute inflammatory response and have enzymes that allow them to destroy engulfed pathogen. All three types of these granulocytes possess receptors for The FC portion, all three types of these granulocytes, you see, possess receptors for the FC portion of the IgG, which is the CD60, and the complement receptor C5a. Now we have CR1, which is the CD35, and we have the CR3, which is CD11b. Additionally, eosinophil possesses a low affinity for the FC receptor for IgE, and therefore plays a critical role in allergic reaction and inflammation in parasitic infection. So let us remember that IgE is usually associated with anaphylactic allergic reaction. So those kind of reaction we can expect a mountain of IgE. Basophil now and mast cell, a type of tissue basophil, possesses high affinity for the FC immunoglobulin E receptor, are powerful effectors of inflammation and allergic reaction, and can cause the release of localized histamine. So this now is where we are seeing the situation becoming a little bit more intense. So both the eosinophil and the basophil are associated with allergic reaction. Lymphocytic cells are generated in the thymus or the bone marrow, and they travel in the circulatory system, or should we say through the circulatory system, to the lymph node and the spleen, where they mature and differentiate. In a mature adult, the lymphocyte accounts for about 20 to 30% of circulating lymphocyte. In primary organs, these cells acquire receptors that enable them to interact with antigens and to differentiate between self and non-self. So our lymphocytes are very important. If we see population increase or decrease in the leukocyte presentation when the smear is done, we have an idea as to the body executing some compensatory mechanism, or the body is in a state of distress. In the secondary organs, the immune cells are provided with a highly interactive environment in which the immune responses are exchanged and made specific. So the immune system in some instances are general, but in other instances it has to be specific. And that is what gives the immune system its robustity. Now, let us move into cytokines and immunoregulator molecules. This is going to be interesting. When we think of cytokines, I want you to think of soluble proteins or peptide molecules that function as a powerful mediator of the immune system. There are two main cytokine types. We have the lymphokines, which are produced by the lymphocytes, because lymphokines, lymphocytes. And we have the monokines, which are produced by the, you got it, monocytes and the macrophages. So the name gives us a relationship. Lymphokines, lymphocytes. Monokines, monocytes. And we know that when they're in tissue, they are called macrophages. So it's the same thing. So cytokines function in a complex manner by regulating growth, mobility, and the differentiation of leukocytes. One cytokine may act by itself or together with other cytokines because there is a power in unity. Other cytokines oppose the action of one or more cytokines and functions to quantitatively increase or decrease a particular immune reaction. So these are necessary when the body is in a compensatory state and it is trying to stabilize and regulate to normal functionality. So some cytokines are synergetic and need each other, while others have their full effect. The effect of the cytokines can be in the immediate area of their release, or they can travel through the plasma to different distant cells and tissue. There is also significant overlap with how the cytokines interact. So the major classes of cytokines include the following: interleukins, that's IL, we have interferons, that's IFN, then we have tumor necrosis factor, that's TNF, and we also have colony stimulating factor, that's CSF, not CSF as in cerebral spinal fluid. But when it comes on to cytokines, we are talking about colony stimulating factor for CSF. To understand the mechanism of action, we need to realize that cytokines act by binding to specific target cell receptors. When the cytokines bind to their receptor on the cell, the number of the receptor is often increased as the cell is stimulated. Internal cellular signaling pathway becomes activated. The cell is no longer in a resting state and can have a new function. After the cytokine bind, both the receptors and the cytokines becomes internalized, which induce the target cell to grow and differentiate. And you know we want the cells to differentiate to go into their specific predetermined function so that specificity can be maintained. Differentiated cells have a specialized function, such as secreting antibodies or producing enzyme. Immune cells and other host cells respond to cytokines and can react with chemoattractant as well as antiviral, anti-proliferation, and immunomodulation processes. So they're playing a role in the fine-tuning of the immune system, which also function as a critical cell activator. So these cytokines are very important to the immune system. Now let us move into immune system genetics. Individuals tend to have unique immune response, just as how they are different individuals. However, in some instances, familial partners, or there can be familial patterns of susceptibility and resistance. In blood banking, this is important because not all transfusion recipients make antibody to alloantigens on a transfused red blood cell. Responders are the people who have the tendency, based on their inheritance, to make antibodies. And when we speak of responders, we consider some to be high responders and others to be low responders, which describes individuals' response to antigen challenge. What we need to realize is that when antigenic stimulation occurs, one B cell forms an antibody with a single specificity. During the lifetime of the cell can switch to make a different isotypic class of antibody that has the same antigen specificity. Isotype switching requires further DNA rearrangement in the mature B cell. The class switching is dependent on the antigenic stimulation and on the presence of the cytokines released by T cells. This reflects the further adaptation of the immune response. An illustration of isotype switching is seen in blood banking when antibodies react at different temperatures and phases. We have been speaking about antibodies, but let us examine the characteristics of immunoglobulins, which are also called antibodies, which is a complex self-protein produced by the plasma. Immunoglobulin is an antibody that is produced by the plasma cells with the specificity to antigens or immunogens that stimulates the production. So when there is a need, there is a stimulation in order to produce the antibodies. So we have immunogens that stimulate the immune system to activate the plasma cells to produce the immunoglobulin. Immunoglobulins act in response to specific foreign non-self proteins or other complex molecules not tolerated by the host. So if it is not something that the host is familiar with or accustomed to or considered to be safe, then that there is going to trigger a response. It's like you have an art piece and you have a security system. So if someone comes into the art theater when the theater is closed or out of service, then there is going to be a trigger of an alarm. Immunoglobulins make up approximately 20% of the protein in disseminated body fluid. Immunoglobulins bind antigens, it fix complements, it facilitates phagocytosis, and neutralize toxic substances in circulation. Thus, we realize that the antibodies have multiple functions, and some more highly specialized and specific than others. But what is important for us to know is that immunoglobulins are characterized or classified into different classes based on their molecular structure of their heavy chain. The 5 classifications are IgA, A representing alpha, which means that this immunoglobulin has an alpha heavy chain. Then we have IgD, which is delta, indicating that there is a delta heavy chain. IgE, which is epsilon, which means that there is an epsilon heavy chain. IgG, which is gamma, indicating to us that there is a gamma heavy chain. Then IgM, which is mu, indicating that there is a mu heavy chain. So to get a good acronym, we can call it GAMED. So that's GAMED, not in any specific order. All right. But if you want to do it sequentially, it is IgA, IgD, IgE, IgG, and IgM. Difference in classification exists based on the molecular weight, percent in serum, the valency or the number of antigen binding sites, carbohydrate contents, the half-life in the blood, and whether they exist as a monomer or a IgG is the most common concentrated in serum, comprising of approximately 80% of the total IgG, next to IgA at about 13%, although it is the majority of IgG found in body secretion. IgM is 6%, IgD is 1%, and IgE is the least common, present at less than 1%. With the different classification, we are expecting that they must have different structures, which would give rise to the variations that we're seeing. All classes and subclasses of immunoglobulins have a common biochemical structural configuration with similarities and derived from a common evolutionary molecular structure with well-defined function. The basic IgE structure unit is comprised of 4 polypeptide chains, 2 identical light chain, the molecular weight approximately 22,500 Daltons, and 2 identical heavy chain with molecular weight from approximately 50 to 75,000 Daltons. And these structures are held together by different bondings. They are not just held together by thin air. They are held together by covalent disulfide bonds, which holds the light and the heavy chain together, and the covalent disulfide linkage in the IgG molecules provide a greater structural strength than do the hydrogen bonds and the van der Waals forces. However, these limit the flexibility of the immunoglobulin molecule. The heavy chains are also interconnected by the disulfide linkage in the hinge region, that join region, the opening closing of the molecule. Although there are 5 types of heavy chain, there are only 2 types of light chains. We have the kappa and the lambda. Both types are found in all classes of the immunoglobulin, regardless of the heavy chain classification. As we look a little closer, we realize that immunoglobulin molecules are proteins and therefore have two terminal regions, NH2 terminal and the carboxyl COOH terminal. The carboxyl region of all heavy chains have a relatively constant amino acid sequence and thus is named the constant region. It has constant amino acid sequence, hence it is named the constant region. Like the heavy chain, the light chain also have a constant region. Due to the certain common Ig structure, enzyme treatment yield specific cleavage products of defined molecular weight and structure. The enzyme papain splits the antibody molecules at the hinge where we have the sulfide bonds to give 3 fragments, 1 crystallizable FC fragment and 2 antigen binding fragments, which are considered the FAB. The FC fragment encompasses the portion of the Ig molecule from the carboxylic region to the hinge region and is responsible for complement fixation and monocyte binding by the FC receptors on the cell. The FC fragment on the Igm antibody is also responsible for placental transfer. The FC fragment on the Igm antibody has another responsibility for placental transfer. In contrast, when we look at the carboxyl COOH terminal, this region, the amino terminal region of both light and heavy chain of the immunoglobulin are known as the variable region because they are structured according to great variation. Hence, you have the idea. They have variation, so they are the variable region. As they are structured according to great variation in antibody specificity. The Fab fragment encompasses the portion of the immunoglobulin from the hinge region to the amino terminal end and are the regions responsible for the binding of antigen. The domains of the immunoglobulins are the regions of the light and heavy chain that are folded into compact globular loop structures. The domains are held together by intrachain covalent disulfide bonds. The V region of the domain specifies the variable region, and the C is the constant region. The domains are also specified according to the light and heavy chain. So let us look at an illustration. One half may have VL, which means variable light region. Another half may have CL, that means constant region, each having light chain. And if it had VH, it would indicate to us that it is a variable heavy chain. Now, important for us to know that there are 3 constant domains. You have the CH1 to the CH3. So you have CH1, CH2, CH3 on the heavy chain of the IgA, IgD, and IgG. And 4 constant domains on the heavy chain of IgE and IgM. And these constants are CH1, CH2, CH3, and CH4. The antigen binding or the idiotypic region distinguish one V domain from all other V domain. These are located within the 3-dimensional structural form by the VL and the VH domain together. So this is just a little breakdown for us to understand what is happening. However, we will realize more details of this immunoglobulin structure when we examine immunology and some areas of serology during our discourse. As it relates to blood banking, we must come to the realization that all antibodies are significant. However, the ones which are of most significant includes the IgG, the IgM, and the IgA. When we speak of clinically significant antibodies, we are speaking of the antibodies that react at body temperature, which is approximately 37 degrees Celsius. These are typically the IgG isotypes and they are capable of destroying transfused antigen positive red blood cell, causing anemia and a transfusion reaction of a various severity, which is something that we try to navigate away from, especially if we're trying to help a patient to be able to compensate for the lack they have. When we consider IgM antibodies, they are most commonly encountered as naturally occurring antibodies in the ABO system and are believed to be produced in response to commonly occurring antigens such as intestinal flora and pollen grains. So blood groups such as Lewis, T, MNS may also produce IgM antibodies, which usually reacts best at ambient temperature, which we consider to be 22 degrees Celsius to 24 degrees Celsius. And if we put our mind back in our initial discussion where platelets are stored, this would appear to be the same temperature required for platelet storage for 1 to 5 days, which we consider to be room temperature. As it relates to testing, the primary problem that is encountered is that IgM interferes with the detection of the clinically significant antibodies by masking their reactivity. Unlike the IgG, the IgM exists in both the monomeric and the polymeric form as a pentamer. So the IgM exists as a pentamer, which is basically pent, that indicates to us 5, containing a J region, which is the joining region, which we did not mention before. The pentametric form of the IgM can be dissociated through cleavage of covalent bond, interconnecting the monomeric subunits and the J joining chain, chemical treatment with sulfide reducing agents such as B2 mercapsuletol 2-ME or diethionyl triethyl DTT. These reagents can distinguish a mixture of IgM and IgG antibodies. Treatments with sulfhydryl reducing reagents such as beta-2 mercaptoethanol 2-ME or ditriotrietol DTT. These reagents can distinguish a mixture of IgM and IgG antibodies because only IgM is removed by the use of these compounds. Therefore, the removal allows unexpected IgG antibodies to be detected. IgG antibodies are significant in transfusion medicine because they are the class of immunoglobulins that are made in response to the transfusion with non-self antigens on the blood products. The IgG antibodies are important in hemolytic disease of the newborn, HDN, commonly or formally called hemoblastosis fetalis, because antibodies can be formed in response against alloantigens on the fetal red blood cell that enters the mother's circulation, usually during delivery. IgG has the greatest numbers of subclass. You have IgG1, IgG2, IgG3, IgG4. Yes, it's only 4. All 4 are easily separated by electrophoresis. The small differences in chemical structure within the constant region of the gamma heavy chain distinguish the various subclasses, and the number of disulfide bonds between the 2 heavy chain is in the hinge region of the molecule constitutes one of the main differences between the subclasses. Functional difference between the subclasses include the ability to fix complements and to cross the placenta. IgG blood group antibodies of a single specificity are not necessarily one specific subclass. All four subclasses may be present, or one may be predominant. For example, the antibody of the Rh system antigen are most likely IgG1 and IgG3 subclasses, whereas for anti-K, KEL, and anti-FY, which is Duffy antibodies, they are usually the IgG1 subclass. However, for anti-JK or KID antibodies, they are mainly IgG subclass 3. We should note that, like IgM, IgA exists in two main forms, a monomer and a polymer form, as a dimer or a trimer composed of 2 or 3 identical monomers, respectively, joined by a J chain, just as in M. IgA is located in different parts of the immune system depending on the subclass. The serum IgA is found in both monomeric and polymeric form. However, secretory IgA is usually found in the mucosal tissues of the body. IgA is important in immunohematology because about 30% of anti-A and anti-B antibodies are of IgA classes. The remaining percentage are IgM and IgG. Also, anti-IgA antibodies can cause severe anaphylaxis if IgA are transfused in plasma products to patients with a deficiency in IgA. IgA can also increase the effect of IgG-induced red blood cell hemolysis. Let us touch a little bit on the IgE. IgE is normally found only in a monomeric form in trace concentration in the serum at about 0.004 of the total immunoglobulin and is important in allergic reaction. The question you may be asking, are there any variations, immunoglobulin variations that exist? The answer is yes. There are 3 main types of antibody inherited variations. We have the isotype, the allotype, and the idiotype. The isotype variation or class variation refers to a variant present in all members of a species, including the different heavy and light chains and the different subclasses. All humans have the same immunoglobulin classes and subclasses. The allotypic variation is present primarily in the constant region. So the allotypic variation is present primarily in the constant region. Not all variants occur in all members of a species. Then we have the idiotypic variation, which determines the antigen binding specificity or the complementary determination region, the CDRs, of the antibodies and the T cell receptor, is found only in the variable and hypervariable region and is specific for each antibody molecule. It is stated that allotypes of a growing fetus may cause the maternal immune system to become immunized to parental allotypic determinants on the fetal immunoglobulins. Just as alloimmunization can occur during pregnancy, it can occur from transfusions, especially in patients who have received multiple transfusions of blood, plasma, or gamma globulins. The idiotypes can be seen as non-self because they are often present at concentrations too low to induce self-tolerance. Idiotypes are determined by the antigen that reacts with them and exists in a type of equilibrium with anti-idiotypic antibodies. If we should just move over a little bit and talk about the FC receptors. Macrophages and monocytes have receptors for the attachment of IgGs and can bind the CH3 dominance of the FC portion. Only
the IgG1 and IgG